Hi everyone, my name is Sydney and today I'm going to be talking about my research about the consequences of asexual reproduction on behavior with evidence for trait decay using the model organism Potamopyrgus antipodarum, or Potamos more colloquially. And Potamos is a really special organism in that sexual and asexual lineages coexist, making it a really great model for the study of sexual reproduction. So broadly, my research focuses on trait decay, and trait decay is a common result of natural evolutionary processes. Traits that once served a purpose and were perhaps even adaptations may become useless over time, and they may cost more to maintain than what they're worth. In this event, traits are likely to be lost over time. One common example is the blind cave fish. And this fish has lost nearly all visual structures and no longer has eyes, and this is because it lives in dark caves, so eyes serve no purpose for the fish any longer. Another example are wingless insects. The second group of pictures show the transition from a once winged stick insect ancestor to a now wingless and flightless version of that insect. And finally, snakes have lost their limbs over time, evolving from previously limbed ancestors such as lizards. We also see this pattern of trait loss in organisms that transition from a sexual mode of reproduction to asexual or clonal reproduction. Most asexual organisms no longer require the traits and anatomical structures that are needed for sexual reproduction, and so such traits are expected to decay and eventually to be lost. Additionally, because mating is associated with increased risks and costs, like increased risk of predation during copulation, greater energy expenditure, and a decrease in time and energy available for other necessary activities, it's expected that these increased costs will facilitate and expedite the loss of mating-related traits in asexual organisms. And such trait loss has been observed in several studies. One study using Potamos found that sperm in asexually produced male Potamos are abnormal, indicating that normal sperm production is decaying in asexual males. Another study found that in an asexual stick insect, females are found to have abnormal sperm storage structures relative to sexual females of the same species. And both of these examples reveal how sexual traits can decay in asexual contexts. My research asks whether the same is true of behavioral traits that are related to reproduction. So in this study, I assessed the mate choice behavior of sexual and asexual female Potamos. Previous studies have assessed the mating behavior of male Potamos and have found that sexual and asexual males both lack the ability to discriminate between good and bad mating partners. Here, I assessed the role of female choice, which has been neglected previously. In this study, I used mate choice assays to compare the mating behavior of sexual and asexual Potamos, and these assays allowed me to address the question of whether or not mate choice is a behavior that has decayed in asexual female Potamos. So in my experimental setup, sexual and asexual females made choices between a good and a bad mate. The good mate choice is represented by sexual P. antipodarum males, and this was considered a good choice because mating efforts between members of the same species have the potential to generate offspring, which is obviously the whole goal of mating. The bad choice then was a male of an outgroup species, Potamopyrgus estrinus. These two species are reproductively isolated, and so no offspring can be generated as a result of any mating efforts. Importantly though, individuals from these species have been known to engage in mating behavior, making P. estrinus an appropriate bad choice. I then recorded the sexual and asexual female behavior using time-lapse video software, and I quantified both the mating frequency and duration. So I assessed both the amount of time that females spent with each male choice, as well as the number of times that they attempted to mate with each male, and I analyzed these data using a Wilcoxon rank sum test. I used violin plots to visualize the data, which are very similar to box plots, but they also show the probability density of the data at certain values, and here I've represented the medians with black dots. I found that sexual females mate significantly more frequently with the good choice than they do with the bad choice. Asexual females, on the other hand, mate with the good and bad choice indiscriminately. Additionally, sexual females mate for longer with the good choice than they do with the bad choice, while asexuals again mate with each choice with equal duration. Overall, I found that sexual female Potamos prefer to mate with P. antipodarum over P. estrinus males, and by contrast, asexual female Potamos showed no evidence for a preference for either mate choice. These findings suggest that mate choice is exhibited by sexual but not asexual female Potamos. This indicates that these behaviors have decayed following the transition to asexuality. These results are among the first to show the decay of behaviors in asexual organisms, and they have great implications on the extent to which asexual reproduction can facilitate the evolution of physical and behavioral characteristics. So these results again indicate that mate choice has decayed somewhere along the transition from sexual to asexual reproduction in female potamos. However, the exact evolutionary mechanism of this behavioral change is still unknown. Further studies will be needed to shed light on why these differences are observed and whether neutral evolutionary forces drive this change or if these changes are more cost-driven. I just wanted to thank the members of the Neiman Lab for their continued support, with special thanks to Dr. Maurine Neiman and Joe Jelinski, and also to iCrew for funding and opportunities to share my research. And here are my references. Thank you.